This video is part 2 for subtopic 5.1, Evidence for Evolution, Selective Breeding, Homologous Structures, and Variation and Divergence. The understandings that we'll cover in this video are related to that of selective breeding, homologous structures, continuous variation, and the applications and skills that we will look at will include development of melanistic insects in polluted areas and comparison of the pentadactyl limb. So we will continue to look at evidence for evolution, and we will focus in specifically on homologous structures and selective breeding and how that contributes to that large body of evidence to show that evolution has occurred and is occurring. So with selective breeding, this is the process by which humans breed animals and plants for particular traits. This is done in a planned and deliberate manner. And it will involve and does involve identifying the largest, the best, or the most useful progeny and using them as the next generation of parents. So you are selecting animals or organisms for their most useful traits and purposefully breeding them in a hopes that the next generation will have those similar traits. But it also results in the continuous removal of progeny showing the less desired traits. And so generation after generation, as this is done, this will eventually lead to a deliberate genetic change. And so over time, the genetic constitution of a population may change. So the example here, the first example, and there are many examples of this, involve doves or pigeons, through which various types of breeds or varieties of pigeons now exist. Other examples of selective breeding include breeding dogs for herding, for example, those specific traits and characteristics, or, for example, breeding greyhounds for racing. And there's many different breeds of dogs out there, all with its different types of purpose. Another example of selective breeding includes corn, for example, where corn, started out as a little bit of millet, and traits were selected for to have larger and juicier and sweeter kernels until we have the variety of corn that we know today. And so crops over time have also been bred for increased yield and disease resistance, or disease resistance if possible. Another example includes sheep. So here we have the sheep that we know or would attribute to a typical farm that has nice thick fur. But there are wild sheep that occur in some parts of the world. And as they were bred for their particular traits or more desired traits, there is a particular type of sheep, the soy sheep, which suggests what the earliest domesticated sheep may have looked like. And then continued modern selective breeding, produced the variety of sheep that we know today, which are shorter animals with a woolly fleece. And many of these breeds have also lost their horns. One last example looking at selective breeding is the variety of vegetables that we have, which include broccoli, cabbage, Brussels sprouts, and kale, all derive from the same ancestral species. So these different vegetables basically were all selected from one species of wild mustard, so they all derive from the same plant. And by selecting variations in different parts of the plants, breeders have obtained these very divergent results. So broccoli was obtained by selecting for the stems, cabbage selected for by the buds, Brussels sprouts selected for the buds, and then kale selected for by the leaves of the plants. So the way in which selective breeding works And when we talk about selective breeding, again, we're talking about breeding of animals or plants for particular traits, desired traits, and those may be related to some particular type of task, or these may be for aesthetic reasons. We like the look of it. So if you have your population, and a population has to show a variation in traits, and remember those variations in traits come about from the existence of alleles. So think about, blue eyes versus brown eyes versus green eyes. Those are variations due to those differences in alleles. So populations have to show variation in those traits. And so individuals which do not show a desired trait are not allowed to breed. And that's kind of the end of their story there. The individuals which show the most desirable traits are chosen to breed together. Ultimately, the next generation should hopefully have an increased frequency of the desired trait. This process then, those individuals that show those desired traits are then bred. So this process is repeated for many generations until the entire population shows the desired trait. So you just kind of cycle back here in which the most desired individuals showing the most desired traits are bred. The next generation is then selected for with those desirable traits and continued to be allowed to breed. And over time, you select for these desired traits, or the population will eventually show that desired trait. And so in the background, it just shows an example of that with cats. So cats exist in many different forms and varieties. And this all comes for that selective breeding, whether you want long pointy ears, short curly ears, short tail, long tail, fluffy tail versus not fluffy tail, all kinds of different characteristics, for example. Another form of evidence that's used for evolution is through the study of homologous structures. Homologous structures are anatomical features showing similarities in shape, though not necessarily in function, in different organisms. So it's similar structures, they don't necessarily perform the same thing or same function, found in different organisms or different species. So it suggests that the species possessing them are closely related and at some point derived from a common ancestor. So the example of a homologous structure that you will need to know is that of the pentadactyl limb. So um, the pentadactyl limb, basically like that arm, is found in a large range of animals, including bats, whales, and humans. And in each group, the limbs have the same general structure and arrangements of bones, but one is adapted, but each one, sorry, is adapted for different uses. So it is important to be able to distinguish between what is considered a homologous structure versus an analogous structure. So an analogous structures are basically features of different species that are similar in function, but not necessarily in structure. So they do not derive from a common ancestor. So for example, the wings of butterflies and the wings on birds are considered analogous structures. They are both used for flying, but because they are both used for flying, it does not necessarily mean they have a common ancestor. And in fact, if you look at the evolutionary paths for these different organisms, you'll see that they do not derive from common ancestors, or these structures do not derive from a common ancestor. So the pentadactyl limb is also an example of adaptive radiation, or the different uses of the pentadactyl limb is an example of adaptive radiation. So adaptive radiation is used to explain how organisms diverge into a range of new forms from a single common ancestor. Adaptive radiation can occur if the environment changes or new food sources or new habitats become available. So the pentadactyl limb demonstrates this adaptive radiation in vertebrates. So as new food sources or new habitats became available, A species would have a common ancestor at some point, would have adapted to it, and um, that pentadactyl limb would have changed or adapted to that new environment. So, for example, the pentadactyl limb in monkeys, is used for grasping, whereas in horses, the pentadactyl limb is used for running. In dolphins and whales, their pentadactyl limb is used for swimming. In moles, for example, that pentadactyl limb is used for digging, and in bats, their pentadactyl limb is used for flight. And so the common features of that pentadactyl limb is that they are basically five-fingered or have variations of that. There is a forelimb and a hind limb. So in that upper arm or that forelimb, there is a humerus. In the forearm, there is a radius and ulna. They have a wrist made up of carpals, and they have metacarpals and phalanges as those digits. So, in comparison to the pentadactyl limb of mammals, birds, amphibians, and reptiles, it shows different methods of locomotion, and the adaptation of each one for its function. So, the pattern of bones is similar in all amphibians, reptiles, birds, and mammals. They are all made up of a humerus or some form of a humerus, that ulna, radius, carpals, and tarsals, and those metacarpals, as well as those digits. So the differences, though, can be seen in the relative lengths and thicknesses of the bones. So, for example, that humerus is probably the most dramatic example, or the ulna, in which you have varying lengths and thicknesses depending on what it is used for. And so it is important to note that all vertebrates have these five-fingered hands at the ends of each limb. So if they had limbs at some point, they all have this pentadactyl limb. Another example of adaptive radiation comes from the study of Darwin's finches on the Galapagos Islands. And in a later video, we'll get into more detail about Darwin's finches and the Galapagos Islands. Um, but these finches that Darwin studied had different beak shapes to them, which seemed to exploit different uses or were used for different means here. So in the Galapagos Islands, it's thought that there was an original species that arrived to the island, and over time, many species evolved from this original species, each one specializing in different food sources, so beak shape and length, and to be able to exploit different habitats. And as they exploited these different food sources, they eventually evolved into their own species. So, we have these different beak lengths, which might be used for retrieving insects, beaks which are used for crushing berries or nuts, beaks that are used for obtaining insects out of the ground, beaks that are used for carrying twigs or branches to build nests. So in variance and divergence now, which is what adaptive radiation starts to show, is that in each species there is genetic variation between individuals in a population, and there is genetic variation between populations in different areas. So this continuous variation between populations provides support so that populations have the potential to diverge from each other and can evolve into separate species. So as an example of this variation that can exist, for example, in monarch butterflies, you can have a yellow variation or a brown variation. And so with this continuous variation, where you have these traits that can vary in populations, This over time can allow speciation to occur, new species to arise. And so the way species or speciation can potentially happen is that if two populations of a species become separated and do not allow interbreeding and experience different natural selection factors, there's a lot going on here, they can potentially evolve in different ways. So over time, as these two populations do not interbreed, their characteristics will gradually diverge from each other. So characteristics will become unique to the habitats that they live in, and eventually they will be recognizably different, and they will not interbreed anymore. So the example is the giant iguana lizards on the Galapagos Islands. So there is the terrestrial iguana and there is the marine iguana. The marine iguana has developed webbed feet and a lateral flat tail that allows for swimming, which is very different characteristics from the terrestrial iguanas. Now, with the continuous variation, this can result in what is called gradual divergence. And so, with this, basically, the greater the geographical separation and the longer the populations have been separated, the greater the divergence, or the greater the difference in characteristics that you can see. So populations will gradually diverge over time, and it is natural to see continuous variation across a geographical range. So the example provided here is that of the red grouse and the willow ptarmigan. They were at one time classified as two separate species, but the current scientific consensus is that they are both members of the same species or may even be subspecies. There's still a lot of debate about that. So the red grouse, for example, is found in the northern parts of Great Britain here in Scotland, whereas the willow ptarmigan is kind of spread throughout Scotland as well, and parts of northern England, as well as can be found on the island of Ireland. Now, the red grouse is geographically separated enough that it does not interbreed with the willow ptarmigan and has um, uh, uh, created or developed these different characteristics. So this is a common problem in classification to determine when two populations of one species have sufficiently diverged to become two separate species. It's a question of like, are they actually different species or do they just show great variation? Other examples where this variation has come into play includes the development of melanistic insects in polluted areas. So we'll look at two situations here, this normal environment and environmental change in this particular example, involving a species called the peppered moth. The peppered moth is found in two forms. There is the dark form and there is the peppered form. This is where the name comes from. So in the normal form and the normal environment, sorry, in unpolluted areas, tree branches are covered with pale colored lichens, this type of moth, which provide cover for the lighter colored moth. So this is the normal environment in which the moth would rest on, the type of tree the moth would rest on, and this tree is typically covered in these pale lichens. And so this moth blends in well with this tree branch, whereas the dark form stands out quite dramatically. And so the moths are hunted by birds while resting on trees. That dark form is more easily spotted and eaten more frequently. So you have low frequency of those dark moths. You have less dark moths that survive to adulthood and reproduce. So as a result, that trait is a lot less common. Now, with the Industrial Revolution that came along, this caused an environmental change. So, sulfur dioxide pollution from industrial areas started to land on the trees and killed off the lichens, and the soot from that also started to blacken the trees. So the trees took on this darker form here. So the dark moths now were well camouflaged from birds against these dark trees, and many more started to survive and reproduce. But now the peppered moth were easily spotted. They stood out against this dark tree and were eaten at a higher frequency, and so fewer forms of these peppered moths survived to adulthood to reproduce. And so over a number of generations, the dark form of the moth increased with frequency, and basically this resulted in the heritable characteristics of that population changing. So you saw a greater frequency or population shift away from the peppered form to the dark form in these polluted areas. So that is the end of this video for part 2 of 5.1, Evidence for Evolution.